Photosynthesis is a topic that I don't think gets enough attention and importance given to it when it's introduced to students in biochemistry classes or biology classes, for instance. And the reason for that is if you stand back and ask yourself, how did animals and how did we even come about? Where did all of our food come from? The ultimate source of that food had to have, at some point in time, been created by an autotroph. And that autotroph would have used a mechanism such as photosynthesis to have created that energy. So even if you ate a hamburger that was made of beef and came from a cow, where did that beef come from? That cow had to eat some grass that had to do photosynthesis to make it. And so it's a huge source of where all of our food comes from. And then in addition to that, all of the oxygen that exists on Earth comes from photosynthesis. We wouldn't be able to breathe if it wasn't for photosynthesis. So it is an incredibly important topic to understand and appreciate in the context of biology and life. And so what I'm going to do in this video is give an introduction to photosynthesis and describe the mechanisms behind it. And so the first thing to start off with is to take a step back and look at what we know about cellular respiration already. If we want to generate ATP, we need to somehow capture the chemical energy that is present in a glucose molecule, and we do that by performing glycolysis to extract some NADH and generate a few ATPs by substrate level phosphorylation. And then once we've done that, we'll throw it through the Calvin cycle, generate a lot more NADH and some FADH2 to go through the electron transport chain and generate even more ATP, and ultimately we end up with carbon dioxide and water as byproducts. And what you should see from this picture is that we are breaking glucose down. We are oxidizing glucose, and when we break down molecules, we call it a catabolic reaction. Catabolic means that it will be an exergonic reaction. Energy is going to be released as a result, and our cells are about 40% efficient at capturing this energy that's released in the form of ATP. Now, when we're performing photosynthesis, what's going to happen is we're going to be building up carbon dioxide. We're going to be reducing carbon dioxide, going in the opposite of oxidation, in order to synthesize sugar. And it's going to be an anabolic reaction. Think of anabolic steroids. You're building up muscles, you're building up carbon dioxide in this case to turn it into glucose. And what is going to need to happen, therefore, is we're going to need to supply energy to our system. Energy will need to enter our system. It will need to be an endergonic process. These are non-spontaneous processes, and therefore somehow we're going to be supplying energy to our system in order to drive photosynthesis forward. And if we look at a typical plant cell to the bottom left here, first thing to make a note of that when I first learned it in high school, I thought plant cells just had chloroplasts, but they do also have mitochondria because the plants will use the same glucose that they make from their chloroplasts in their own mitochondria too. So plants do have both chloroplasts and mitochondria, but for the sake of learning photosynthesis, we are interested in chloroplasts. And so if we zoom in on a typical chloroplast, what we would see is this figure to the right here. And the inside of the chloroplast is called the stroma. And you can think of the stroma like the cytosol of a bacteria. And according to the endosymbiotic theory, we know that bacteria were enveloped by a host cell, and this is very much what we see here. And within each thylakoid, we see within each chloroplast, we see stacks of thylakoids, and within each thylakoid, we see something called the thylakoid space. And so these are two terms that you really want to familiarize yourself with, because just like what's going on with mitochondria, we are creating a proton gradient inside of these chloroplasts. What's going on in photosynthesis is we are pumping protons from the stroma into the thylakoid space. Thylakoid space is just the inside of the thylakoid. And we have an electron transport chain exactly like we had in the inner membrane of the mitochondria. And so if we continue our discussion and look more at the chemistry that's going on here, what we're going to see is that we have photosynthesis being broken down into two parts. We have light-dependent reactions, which would be this left half, and then we have the dark reactions, which is also called the Calvin cycle, which makes up the right half of this image to the bottom left corner. And the key intermediates that we see in this reaction are NADPH, NADP+, ATP, and ADP. And then what is important to make a note of about NADP plus relative to NAD plus is it is the exact same molecule except for the existence now of this additional phosphate group here. And the purpose of this phosphate is to give NADP plus specificity for anabolic enzymes. So when there's a lot of NADPH, the other form of NADP plus, once it's taken the electrons and the proton, it's going to need to work with some anabolic enzymes in order to help synthesize glucose molecules in the dark reactions. And so learning the structure and seeing the differences and also the similarities is very important. And then also to talk a little bit about what's going on in the light-dependent reactions. So if we were to zoom in our thylakoid membrane, in our chloroplast, we'd see structures that look much like what we see here. And what these things are referring to are these photosynthesis complexes, and P680 refers to a particular protein complex that can take in a 680 nanometer wavelength photon, and it will cause that 680 nanometer photons energy to excite an electron that exists on the P680 complex. And commonly you have a magnesium or an iron atom present, and this is because metal ions are very good at holding onto one electron. And if you have a background in organic chemistry, what amazes me about photosynthesis is the fact that we're going to be taking electrons away from a water molecule. And what we know about water from organic chemistry is that water is an extremely unreactive substance. And so to be able to just take some electrons away from water is incredible to me. So it says a lot about this P680 complex. And so intuitively, if you want to know a little bit more about what's going on here, so the electrons that exist on P680, when a photon strikes it that is just the correct wavelength, the electrons will jump into a higher n orbital, into another shell, a higher energy shell. And when the electrons exist in this high energy shell, they're a lot more excited, and P680 goes from P680 to P680 plus. And P680 plus has a very low electrophilicity to it. It really doesn't want those electrons anymore. It really wants to give those electrons onto something else. And so if you take physical chemistry or quantum mechanics, you can much better understand what is going on at this critical step that makes photosynthesis possible. And plants have been doing this for millions of years. But the gist of it is we are seeing a higher orbital or a higher shell realized by the electrons. And when we are analyzing the processes that are going on in our thylakoid membranes, what helps a very key term to learn is something called reduction potential. And what we know about electricity, it has units of volts. And in the case of reduction potentials, more positive equals more electrophilic. So if you have a very positive reduction potential, it means you really want your electrons. And what we see with the water here is water has a very high electrophilicity. Note how the axis, when you read these plots, are typically inverted. This one is inverted, so down this plot really means it has a more positive reduction potential. But that is a very key point to understand behind the intuition of these complexes. And then also a final note I'll make about what is going on in these complexes is as these protons move through these electron transport chains that exist in your thylakoid lumen, or the thylakoid membrane. Sorry, it is the thylakoid membrane. As the electrons travel through, what we essentially see within each one of these protein complexes is a series of protonations and deprotonations, and that is a consequence of the electrons moving through it. Because the electrons are carrying a negative charge, the consequence of that negative charge is that a protein can be protonated or deprotonated as the electrons move through it. And if what we're wanting to happen is the deprotonations to occur within the thylakoid lumen, because we are wanting to pump protons into it to establish the proton gradient in order to drive ATP synthase and generate NADPH from NADP+. And so that is another piece of intuition that I think would be very useful in an initial discussion of photosynthesis. And so this is going to wrap things up. I'll probably make some more videos about photosynthesis because it is a very important subject to understand in biology and biochemistry. And I hope you guys find this useful. Let me know if you have any questions, and thanks for watching.